Hello, everyone. A very big good evening to you all. Good evening, all the students. Am I
Hello
audible?
everyone. A very big good evening.
Am I audible, everybody? You can just send some reply in the chat box. Hello. Hi, everybody. So, myself, Tithi here, and welcome to this session of Brahmastra Series. And in today's class, we are going to talk about mutation, Unit 8 mutation. Now, from Unit 8, there are many important topics. Unit 8 is for genetics, and from that, there are many important topics for your CSIR syllabus. But mutation is also one of the very important ones. The reason for it being important is because this concept is related to other concepts of other units also. Okay, and nowadays, like if you see the questions which are asked, so they are more combination kind of questions which combines the concepts of multiple topics or multiple topics from different units as well. Okay, and mutation is one such topic which is which is like a connecting link which will be there between different topics of different units. So mutation, what are the different types and all you study in genetics. Then the different diseases, genetic disorders which are caused by mutation, that also we study in unit 8. Along with that, how to detect mutation, those comes in the methods of biology, that is unit 30. Then the mutation, so genes are always there, right? So genes which help in development, and also developmental biology is there. So if there is any mutation happening in those genes, what kind of changes will happen, that you study in developmental biology. Then mutation is a major cause of cancer. So in cell programming, cell death also, you study the different effects of mutations, different genes, and what their mutations, how their mutations, what kind of mutations can arise in them, and how it will lead to different types of disorders or different types of cancers. And then mutation is also related to molecular biology, right? So when you study the DNA repair mechanism, so why there is repair, because some change has happened, some mistake has happened during DNA replication, or by some external agent, some change in the genetic material has happened, so the cell has to repair it, okay, or cell has to correct it. So what that mistake is, that mistake is nothing but mutation. So the repair enzymes are working to detect and to rectify that mutation. Okay, so that is what telling you, that is why I'm telling you that this mutation is a topic which is linked to multiple topics in the syllabus, and that's why it is extremely important. And the best part about mutation is that this topic is not very difficult to understand. There is no mathematics as such involved, which usually biology students run from. And the concepts are also kind of simple, but basic concepts have to be strong here. Your concepts, as you know, for entire CSI syllabus, your basic concept have to be strong. Excuse me. And that is when you will be able to solve this question from mutation very easily. Okay, so comparatively it is an easy topic, and it is important at the same time, so that's why you can never leave this. Now I hope all of your preparation is going well. Yes. Can I have some quick chats from you all? Can you just send some hi? Or if you are ready for me to start the question discussion about BPOs, bring it on. Hi Krishna. Thank you so much for sending message. So I was just wondering whether the message service is working or not. Okay. All right. So are you all ready? And come on, guys, let's have an interactive session. So, after showing every question, I'll give you a couple of minutes for you to analyze it, and then we will see the solution here. Also, I will show you the solution, but I want you all to try to solve it as well. Okay, then, and please make it interactive. All right, that part is up to you. You people have to continuously send me messages. Okay, then only I will know that you people are there. Okay, all right, Krishna. So let's get started. I'm very sure, like Krishna, there are other students also. Please respond in the chat box, everyone. Okay, very first question is here. Now, I'm very sure that all of you are preparing for the February exam. So you all have started practicing previous year questions. So if you are practicing previous year question, this type of question, I'm very sure you have come across, because this is a very popular kind of question which is very frequently asked. It's a very popular kind of question. If you take 10 years question papers from 10 years back, that time also you will find this question. If you take question paper from 5 years back, then also it is there. And also, like this session, June session paper also, if you take, then also this question was there. So this is a very, very popular kind of question. Good evening, beautiful colors. Welcome to the session. Good evening, Varinder. Good evening. Yes. So I want you all to analyze and try to answer this question. The figure below, given below, is of two homologous chromosomes paired during meiosis, where one event of recombination occurred between the two homologs. The following interpretations were made. So A, B, C, D, E, five interpretations are there. Which of the options given below has all correct answers? Yes. What do you guys think? What should be the answer? Slide is not clear, then please go to the setting option, Varinder, and you can choose what kind of setting you want in your during the session. Okay, so just choose the 720 HD one. Go to the setting option. Yes, and our technical team has displayed the process also right below the below the video. It is coming. Just follow it. Okay. Right. So let's have a look at this question. Everyone, please pay attention. Okay. So this is a kind of question where, first of all, you have to determine whether it is a pericentric inversion or paracentric inversion. Now, before that, yet, let me tell you, like, from where this question is. So the question is coming from inversion. So what happens is, when there is inversion in a heterozygous condition, means only one chromosome is having inversion, the other chromosome is not. Okay, so in that case, at the time of meiosis, when the homologous chromosomes have to align, so straight, just like this, the chromosomes cannot align. Why they cannot align? Because the segments are not matching. So suppose this is CDE and this one has got inversion. Okay, so can they align like this straight away? No, because this part is not aligning. Okay, so in that case, how the alignment happens is that one of the chromosome will make a cross loop. Do you see this blue one is making a cross loop, and that is how the alignment will happen. Okay, now usually this making a cross loop is a strain that is there on the chromosome. So usually, if there is heterozygous inversion, then crossing over does not happen through the inverted segment. Okay, but still, usually it doesn't happen, and that's why crossing over is also like inversion in heterozygous condition is also called as a suppressor of crossing over. But if by chance still it happens, okay, then what will be the consequence? So this question is related to that. Okay, so whenever you see question like this, a structure involving this, that means it is talking about a heterozygous inversion. Okay, now what will be the consequence of crossing over occurring through that heterozygous inverted segment? That will depend on Good evening, Vrinda. Yes, you are absolutely correct. Yes, so that is what I was saying, that the consequence of it will depend on what kind of inversion it is. Okay, so first thing you have to detect what kind of inversion. All right, now what kind of inversion it is? So we know that inversion is of two types, pericentric and paracentric. So since the inverted segment, now how do you recognize which one is the inverted segment? The segment which is coming inside the loop. Okay, so that is the inverted segment. So this inverted segment, since it does not include centromere, then it is a paracentric inversion. Okay, so now you know that this is paracentric inversion. All right, now if it is paracentric inversion, then what will be the consequence? As I am telling you, that consequence will vary depending on whether it is a heterozygous paracentric inversion or heterozygous pericentric inversion. Okay, so let's have a look here. So individual in yes, in case of pericentric, sorry, in case of paracentric inversion, as you see here, so when there is paracentric inversion, okay, so you see the same structure is given here also. Okay, so when crossing over happens like this, then it will result in the formation of a acentric fragment and a dicentric fragment. Okay, now this acentric fragment will be lost during the first cellular division, because this first meiotic division, because there is no centromere, so there is no way of spindle fibers attaching to it. Okay, now what will happen to this one, the dicentric bridge? Now for this dicentric bridge, so this chromatids will be pulled towards opposite pole. So when they are pulled towards opposite pole, so this dicentric chromatid will result in the formation of a dicentric bridge, but pulling will continue. So ultimately, like if you have a rubber band and keep on pulling it to the opposite end, what will be the result? The rubber band will break, right? So in the same way, this chromatid will break, and this break will happen abruptly. All right, and so at the time of second meiotic division, suppose it got broken here. Now second meiotic division, the chromatids will separate. Okay, so as you see here, that all these four chromatids will go to separate gametes. The gametes receiving these two chromatids will be functional, will be normal, because they have all the genes. Okay, but the gametes which are receiving this chromatids that participated in crossing over, they will not be non-viable or inviable, because there is deletion of genes. Okay, so let's see the statements one by one. A says that individual is heterozygous for an inversion. That is correct, because only one homologous chromosome has the inversion, the other one does not. So this is correct. Next figure depicts a paracentric inversion. That also we have found out that it is paracentric, because inverted segment does not include centromere. So B is also correct. Okay. Now, as I always tell you, that you should solve question in part. Yes, yeah, Vrinda, your answer is right. Okay, so as I always tell you, that you solve a question in part. So you have determined that A and B is correct, right? Now, if you check the options, so automatically option 2 and 3 are eliminated, because A and B are not included in them. Okay, option 2 has B but doesn't have A. Option 3 doesn't have A or B both. Okay, so you can eliminate them. So either A and B only, or A, B, C, D, E. Okay, now check C. After recombination at anaphase 1, a dicentric and acentric chromatid will be formed. Now that is absolutely correct. That is what happens in paracentric inversion. So C also is correct. Okay, now as soon as you determine that C also is correct, so you can eliminate option 1. Okay, and you know your answer is option 4. All right, see, this way you can save much time in solving. You don't have to waste time in analyzing these two statements altogether. Okay, so this is a trick that you can follow for solving question from any part of the exam, and in some cases it might not work. Okay, you have to analyze the entire question, but in some cases this will work, where it will save time. So wherever it is working, it is saving time and giving you clear answer. Okay, but you can eliminate an option. So there is a catch here, that you can eliminate an option only when you are 100% sure it is not working. Okay, or it cannot be possible. Okay, now what is D saying? At anaphase 2, the recombinant chromatids will have large deletions or duplication. Now ideally this part is not correct. Okay, they will not have duplication, they will have large deletions. Okay, But do you have any option of A, B, C, and E? Because statement E says that inversion are often considered as crossover suppressor because crossover products do not survive. Okay, this also is true. So actually, the correct statements are A, B, C, and E. D is slightly incorrect because of this duplication mentioned here. Okay, but since we do not have any option like that, A, B, and A, B, C, and E. Okay, if this was the option, then this will be the most probable answer. Okay, but because we don't have any option like that, so we have to go with option 4, which is A, B, C, D, and E. Okay, this kind of call also you might have to make during the exam. Okay, so you have to choose an answer which is most correct. Okay, if this kind of situation comes. Right, moving on to next question. Fertilization of gametes containing chromosome with duplication or deletion often result in children with disabilities. What is the probability of a couple where male is karyotypically normal and female has a pericentric inversion in heterozygous condition producing a child with disability if crossing over took place within a pericentric inverted segment in 26% of the meiotic divisions? In this case, consider that fertilization with gamete containing chromosome with duplication or deletion will result in disability. Yes, can I have some answer from you all? So this question is also very much like the previous question only. Okay, situation is same. How the situation is given to you in the question, that is what is different. So in the first one it was all statement based. Okay, and this one you have to do little bit of calculation. Okay, but the concept is exactly same. Very good, Vrinda. Good. What about other students? So this shows, Vrinda, that you are practicing previous year questions. You are doing a good preparation. Keep it up. This time is like the last lap of your preparation. So you should not slow down, just keep on increasing your pace. Okay, so this question, let's see here, everybody. Now I want more answers from you all. So in this question we have, it shows that there is pericentric inversion. Okay, now what will be the result of heterozygous pericentric inversion? Now, just like how we saw in case of paracentric inversion, the pericentric inversion means the inverted segment includes centromere, as you can see here. Okay, now if pericentric inversion is there and crossing over happens through the pericentric inverted segment, then it will result in the formation of, again, the recombinant chromatids will be having mutation. Okay, what kind of mutation? They will have deletion or duplication. Okay, so because of that, the gametes which are receiving this chromatids, they will be inviable because there is deletion or duplication. Okay, so if crossing over has happened, then 50% of the gametes will be inviable because of deletion or duplication. Okay, now in this question it is saying that it is not producing, this deletion or duplication is not producing inviable gametes, but it is producing a child with disability. Okay, and question is, if 26% of the meiotic division there is there is crossing over occurring through the pericentric inverted segment, then what is the chance of producing a child with disability? Okay, so when will the child will have disability? When this recombinant gametes are coming. Okay, so if in one so in one meiotic division, if there is one meiotic division, then 50% of the gamete form will be having the inversion, right? So if there are 26% meiotic divisions where crossing over has happened through through the pericentric inverted segment, then how many gametes will be inviable, okay, or will be having the having this deletion or duplication. So it will be 50% of 26%. Okay, so basically it will be 26% divided by 2, which will be 13%. Okay, so 13% will be the answer. Okay, so I hope all of you understood. Did you all follow? So concept is same, idea is same. The previous question was statement based, this one is mathematical. All right. So before going ahead, yes, this is our Lakshya batch, everyone. All of you know that Biotecnica's Lakshya batch is going to start. It has actually started from today, but admissions are still going on. If you are interested for joining the elite coaching of Biotechnica, please join our Lakshya batch. And you can, if for information, you can call this toll free number. And if you enroll with us with the for the Lakshya batch for June 25 and as well as gate 2026 exam, then along with this coaching you also get molecular biology internship completely free. Okay, so I hope all of you have an information about it. Still, if you want more information, please do not hesitate and you can dial our toll free number for more information. Okay, one more thing, everyone, as you all know that we have some extra questions along with explanations, and those questions will be distributed to the participants at the end of this session. So if you are interested to get those questions and also along with the explanation, please stay with us till the end. Okay, next question. Now, this is also another type of question which is quite popular. Okay, very frequently it has come in the exam. Anybody has the answer? Vrinda, what about you? Other students, I'm waiting for your answer. Okay, let's try to solve it together then. So, an individual is heterozygous for a reciprocal translocation as shown below in the given diagram. Following statements are made about the segregation of such chromosomes during meiosis and gamete formation. So, the complexity paired. So, here what do we see? That yeah, so in here we see that there is a there are four chromosomes, okay, and they have undergone heterozygous reciprocal translocation. Okay, so N1 and N2, these are the two normal chromosomes. T1 and T2 are the ones which have undergone reciprocal translocation. Okay, so following statements are made about the segregation of such chromosome during meiosis and gamete formation. Okay, now when heterozygous reciprocal translocation happens, so even without considering the complexity of crossing over, so the chromosomes will align with each other like this. Okay, so because the translocated chromosomes share parts with both the normal chromosome, so they will form a cross kind of structure. Okay, where the translocated chromosome will align with both the normal chromosome. T2 is aligning with N1 as well as N2. T1 is aligning with N1 as well as N2. Okay, like this the alignment will happen. Okay. Now, once this alignment is done during meiosis 1, there can be three different ways the chromosomes can segregate. Okay. One way is adjacent one, where these two chromosomes go together, these two chromosomes go together. Okay. The other way of segregation is in this way, where these two chromosomes go together towards one pole, these two go towards another pole. Okay. Now, in both of this situation, adjacent one and adjacent two segregation, it will result into inviable gametes. Again, why? The reason being that the gametes will not get a complete set of genes. There will be some genes missing and some genes present in more than one copy. Okay, that because of that it will be inviable. But there is one way of segregation which can still result in the formation of viable gamete, and that is called as alternate segregation. So what happens during alternate segregation? So during alternate segregation, these two chromatids will go together, means both the chromosomes having the translocation, they will go together, and the chromosomes having no translocation, they will go together. Okay, so that will result in the viable gametes because those gametes will have all the genes in their correct number of copy. So let's see the statements given here. Complexly paired four chromosome fail to segregate, pass into one cell at anaphase 1, and the cells eventually die. This is incorrect because they have a way of segregation. Fail to segregate is incorrect. Statement B, chromosomes pair between regions of maximum homology, keeping the translocated part unpaired and a normal meiosis occurred. No, the translocated part also pairs, but not with this chromosome, not with the normal chromosome, but with the chromosome with which it shares the translocated part. Okay, so this is also incorrect. See, one of the ways the chromosome segregate is by alternate segregation. N1, N2 move to one pole, T1, T2 moving to other pole. Now, this is absolutely correct. D, alternate segregation produces non-viable gametes. Incorrect. It produces viable gametes. E, reciprocal translocation are considered as crossover suppressors as no gametes with crossover products are produced. Now, this is also incorrect. Okay, because crossover products, even if they are they are there, but because here we are not considering even crossing over. Okay, we are not considering crossing over here. This is the simple segregation of the reciprocal translocated chromosomes. No crossing over is considered here. Okay, inversion are called as crossover suppressors, not translocation. Okay, so answer will be only one statement is correct, that is option 2, which says C only. Everybody, did you understand? All right, moving on to next question. Please go through the question, try to comprehend it. So if anyone of you can solve this question, you're most welcome to send the answer. Krishna, Varinder, Vrinda, beautiful colors. All right, I hope all of you have read the question. So let's see together. 15 spontaneous ARA minus mutants of E. coli that were not able to utilize arabinose as a sole carbon source in minimal synthetic medium at 42 degrees Celsius were isolated. However, interestingly, all these mutants were able to use arabinose as a sole carbon source at 30 degrees Celsius. Based on the above information, which one of the following option represents the most likely type of mutation? Okay, see, first of all, the information that is given. So basically, what it is saying that E. coli at 42 degrees Celsius can't use arabinose. Okay, but at 30 degrees Celsius, the same cells can use arabinose. Okay, so if randomly, if you think about what kind of mutation it is, so this is a no, Vrinda, that's not correct. What kind of mutation will this be? This is a conditional mutation. I hope all of you know about what is a conditional mutation. A mutation which will produce the mutant, the wild type phenotype during permissive conditions, but during restrictive conditions, it will produce a mutant phenotype. Right? So, at a glimpse, if you see, it is talking about a conditional mutation. Now, when you check the option, is conditional mutation given? No. Okay. So, conditional mutation is there, but basically, if you see the options, deletion, inversion, frameshift mutation, missense mutation, so it is asking about what is the molecular basis of that particular conditional mutation. Okay. So, based on the above information, which of the following option represent the most likely type of information? Okay, we don't know which one is exact mutation that has happened. For that, we need to do more analysis. Okay, but which seems to be the out of this four, which seems to be the most likely type of mutation. Okay, so here is what you have to do the analysis based on the information. The background information you need is information about what are these different mutations, okay, and what will be their consequences. Okay, so when we talk about deletion or inversion, they both are chromosomal mutations, and in this cases what happens that a large segment of DNA is affected, multiple genes are affected. Okay, so if a segment is deleted completely, then if we call it as deletion, so it is completely deleted. Okay, so if it is already deleted, then again at 30 degrees Celsius, how come they are using arabinose? That means the gene is not deleted, just that the gene is working at 30 degrees Celsius, but it is not working at 42 degrees Celsius. Correct? So deletion cannot be there. What about inversion? So when there is inversion, so during inversion, so suppose you have this. Okay, so when you have done inversion, so let's say that these are the different genes, 1 2 3, and this is our ARA gene. Okay, and 4 5. Okay, so when this, suppose this particular segment is inverted. Okay, so when it is inverted, the gene changes its position. Okay, now if the gene is changing its position, then if there is any change in the expression of that gene because of change in position, that is what we call as point effect, position effect, sorry. Right, now if there is position effect because of inversion, if the gene's position has changed and because of which it is not working anymore, then at 30 degrees Celsius also the same position is there for the gene, then how come it starts working again? Correct. So not possible. So inversion also not possible. What about frame shift mutation? Now, frame shift mutation, if it happens, we know that it does not only affect the codon where it is happening, but it affects all the subsequent following codons. Okay, so a big portion of the protein gets mutated, and that is why we say that this frame shift mutation usually happens because of nucleotide insertion or deletion mutations, and that have major effect on the overall structure and function of the protein. What about missense mutation? How does missense mutation happen? Missense mutation happens by base substitution. Okay, and in base substitution, what is the consequence? Only one codon is affected. Right, so when there is base substitution mutation, so that a coding codon changes to a codon that is coding for something else. Okay, the sense is changing, that is what we call as missense mutation. Now in missense mutations, because a single codon changes, so a single amino acid changes. Okay, now if that single amino acid is changing, then it is possible that mutation is caused because of that single amino acid change, and because of that the protein the gene remains expressive at 30 degrees Celsius, but at 42 degrees Celsius it stops working. Okay, because as the temperature is increasing, that particular amino acid which has changed is modifying the structure of the protein even more, so that it is not working now at 42 degrees Celsius. Okay, so and this missense mutation also, it is the least amount of change or least amount of mutation that is happening here. Okay, so that's why answer will be missense mutation. Most likely type of mutation seems to be missense mutation only. Am I clear, everyone? Okay, so it is most likely, again I'm telling you, it is most likely, we cannot say definitely it is missense mutation only, because sickle cell anemia also happens because of a missense mutation, but it has a huge implication. It completely changes the function of the hemoglobin molecule, right, even though it is a single change. Okay, but most likely out of all the mutations that are given here on the option, missense mutation seems to be the most likely answer. Excellent, moving on to next question. Yes, please take your 2 minutes to analyze this question also. And in the meantime, if you get an answer, please let me know. Excuse me. That is very good, Vrinda. Very good. So, can you tell me what concept you have used in solving this question? Yes, Vrinda. Yes, very good. Correct. Anybody else got the answer? Anyone else would like to answer? So as Vrinda just told the class that this particular question can be solved using complementation test or complementation analysis. Okay, so I am telling you again that this is again a proof that how mutation can be linked with other concepts. So complementation test you might have studied as a part of deviation from Mendelian principle, but there also when we are using mutants, when we are creating mutations, and then we are trying to check how many genes are involved or how many complementation groups are there, in how many genes mutations are happening. So there we are actually checking about mutation. Okay, so this concept can be coming in in mutation topic also. So the question here is, and this complementation test is again a very popular topic for CSIR exam. Time and again question has come from here. Okay, but one thing you have to remember, that it is not that in the question they will mention that it is complementation test or a complementation test is done. That will not be mentioned. Okay, so what are the clues to identify that it is a complementation test related question? First of all, a table like this. Okay, if so, in this table what is happening, cross is happening between different pair of mutants. Okay, cross happening between different pair of mutants is a biggest clue for complementation test. Okay, now this thing, the same thing, okay, whether it is given in the form of table or mentioned in the question in the form of statements, okay, that is what you have to identify. that mutants are crossed with mutants in different pairs. So if that is there and then we are analyzing the result of that cross, means we are doing a complementation test. Okay. So in the question, uh, P. aeruginosa makes a blue-green pigment called pyocyanin. To understand the pyocyanin biosynthetic pathway, mutants which cannot make pyocyanin were isolated. Six such mutants were crossed with each other. Okay, mutants crossed with each other, and the data is given below. Minus means pyocyanin negative, positive means pyocyanin positive. Based on this data, can you predict how many genes are responsible for the pyocyanin production? Okay, so this is complementation test. Now, in complementation test, how do we analyze? So, mutant cross with mutant. Okay, then we check the result. So, if we do mutant cross with mutant and the result is wild type, F1 is wild type. Okay, means complementation happened. Okay, and complementation happened will be shown by positive, because positive is showing pyocyanin production. So wild type phenotype is production of pyocyanin. Mutants are not able to produce pyocyanin. Okay, so when mutants are crossed and wild type is produced, okay, means complementation happened, and complementation happened is shown by plus. Okay, so if complementation is there, means the genes are non-allelic in nature. So the mutations, okay, they are, even though the mutations are producing a mutant with same with similar phenotype, but still mutations are occurring in different genes. So if wild type phenotype is produced on crossing mutant with another mutant, that means complementation has happened, which again means that these mutations are in different genes. Okay, mutation in different genes will complement. Okay, if mutant cross with mutant and you again in F1 get mutant phenotype, means complementation has not happened. Okay, and why complementation has not happened? Because the mutations were there in same genes. Okay, now the question is, based on the data, can you predict how many genes are responsible for pyocyanin production? So basically you have to find out how many complementation groups are there, and whatever will be the number of complementation groups, means those many genes are responsible for pyocyanin production. Okay, now when we check the table here, so okay, see, this entire table is given, but you need only half of the table. Other half is just a mirror image of the first half. Okay, see, what is this block? This block is showing 2 crossed with 1, right? What is this block? This is showing 1 crossed with 2. Okay, so whether you cross 1 with 2 or 2 with 1, your answer will be same in F1. Okay, right? So you have to consider only half of the table. Other half is there only to confuse you. Okay, so let's say that we are not considering this half. Okay, we are considering only this half, right? Now when you are considering this half here, okay, you see minus 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 means no complementation is happening. Now definitely it will be no complementation, because this is for what? 1 cross with 1, this is 2 with 2, 3 with 3, so same mutations only you are crossing, so definitely there will be no complementation. Okay, now you want to check among them 1 2 3 4 5 6 who all are not complementing each other. Okay, why not complementing each other? Because they are the one who will be present in the same gene. Okay, so if you see 1, okay, so 1 is plus 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 plus, it is complementing 2 3 4 5, but it is not complementing 6. Okay, what does it tell you? That 1 and 6, they have mutation in the same gene. Remember, mutant cross mutant, F1 mutant means happening in the same gene. So 1 and 6 are happening in the same gene. Okay, what about 2? If you see 2's column, so 2 is complementing all the mutations. Okay, means 2 is happening in a separate gene. Again, if you check 3, 3 is complementing all again, so 3 is again a different gene. If you check for 4, 4 is also complementing all. Okay, If you check for 5, 5 is also complementing on. Okay. So, and 6, we have already placed it with 1. So, how many genes are involved here? 1, 2, 3, 4, 5. So, even though we have considered 6 mutants, mutant 1 and mutant 6 have mutation in the same gene. So, for pyacinin production, there are 5 different genes are involved. Okay. Answer will be option 3. That's correct, Abhijit. Okay, everybody followed. So this complementation test related question can be can come in any format. Okay, but you have to understand this clue. Okay, mutants are crossed with mutants and the F1's phenotype is analyzed. All right, so when you understand this, when you pick up on that clue, for sure it is a complementation test related question and you know how to solve it. Okay, now coming to another very popular kind of question from mutation. Again, this is a question which was popular 10 years back also, 15 years back also, as well as now also. Okay, time and again, this type of question has come. So this question, it is related to um, this reversal test, right? Mutation reversal test. Yes. Anybody would like to tell me what will be the answer? Yes, Vrinda, Abhijit, any other student also, if you want to reply? Excellent, Vrinda. Very good. Yes. So a scientist synthesized 4 new chemicals which had mutagenic potential and named them as C1, C2, C3, and C4. He tried to analyze the nature of mutations caused by them and obtained the following result. Mutations was produced by mutagen C1, C2, C3, and 3,4. So then the mutation reversal study was done. So in mutation reversal study, what do we do is any mutation that is being produced by an unknown chemical, we try to reverse it with a known chemical. Okay. So if reversal has happened, so we know the chemical with which we are doing the reversion. So we also know what kind of mutation that particular mutagen can cause. Okay, so whatever is the mutation that is reversing it, that helps us in identifying the mutation which has caused it in the first place. Okay, so for this type of question, this table will be there. So first of all, you have to recall what is the function of each of these mutagens that are used in the reversion. So we have 2-aminopurine. Okay, 2-aminopurine is a base analog. Nitrous acid, it's a deaminating agent. Hydroxylamine, it's a hydroxylating agent. And acridine orange is an intercalating agent. Okay, now all three of this results into transition. They cause transition mutation. So basically, any mutation which is getting reversed by this three mutagens means it is a transition mutation. Acridine orange, it results into single nucleotide insertion or deletion. Okay, so any mutation that is getting reversed by acridine orange will either be a single nucleotide insertion or deletion. Okay, now in C1, what we see is it is getting reversed by to aminopurine and nitrous acid, and some are getting reversed by hydroxylamine. Now, why there is some? Because hydroxylamine cannot work on all the nucleotides. It can only work on cytosine. Okay, so that's why it will, in some of the cases, it is getting reversed by hydroxylamine. Okay, but 2-aminopurine and nitrous acid, it is reversing, means C1 or mutation caused by C1 has to be a transition. Okay, now you can check the options. So C1 as transition is there in 1. Option 2, eliminate. Option 3, eliminate. And actually, if you see, option 4 also is eliminated, because option 4 is also saying that C1 is transversion. So do you see? You can solve this question by only solving what mutation C1 is. But this is important that you are completely confident about this answer. Okay, so C1 will be transition here. Next, in C2, it is not getting reversed by any of the mutation. Okay, means it will be neither transition nor insertion or deletion. Okay, so what can C2 be? So option 1 says that C2 can be transversion, yes, which is possible, or it can be a large deletion. So acridine orange can only reverse single nucleotide insertion or deletion. If it is large deletion, it cannot work. Okay, so this seems correct. C3 again is getting reversed by this 2, means C3 is again a transition. C3 is transition, and C4 is getting reversed only by acridine orange, means it has to be a single nucleotide insertion or deletion. Okay, answer is option 1. So everyone, I will again request you that this is the high time where you start solving questions, and when you are solving question, please try the sequential elimination process and let me know whether it works for you or not. Okay, so you can eliminate the options, you can solve the question in part, you don't have to solve the entire question and check for options. This strategy says that you solve the question in part, after solving each part check the option and eliminate the option which is not at all possible. Okay, just try this and see whether it works for you or not. If it works, then definitely practice it so that you can apply it during the exam. Okay, now coming to the next question. Okay, so we'll go a little fast here because we have 7 minutes left. Please read the question, everyone. And everybody, just 7 more minutes left. I will request you all to stay till the end. Then only you will be able to get the document with all more questions from the for your practice from this topic of mutation. And the explanation of those questions are also present in that document. So please stay there till the end. I'm sorry, Vishal, that's not correct answer. Okay, so let's have a look at this question. Red blue color blindness is a human X linked recessive disorder. Okay, even though this information is not given to you, you should know because this is such a popular genetic disorder. The two parents with normal color vision have two sons. Son 1 has 47 chromosomes with XXY chromosome composition and is colorblind. Okay, so what do we know? Son 1 is XXY and he is colorblind. Son 2 has 46 chromosomes, so son 2 is normal. Son 1 has trisomy, okay, but son 2 is normal and he also is colorblind. Okay. Assuming that no crossing over took place in prophase 1 of meiosis, Klinefelter syndrome in son 1 resulted due to non-disjunction during which one of the following events? Okay. Now see, it is very clearly mentioned that the gene for color blindness is a X-linked recessive, X-linked recessive. Okay. So suppose color blindness is caused by this gene XC. Okay. So the allele X capital C will be normal, whereas the allele X small c will cause color blindness. Okay. Now, this son, who has the Klinefelter syndrome, which is trisomy of sex chromosome, is colorblind. Okay. Now, this son has two X chromosomes, right? So, still, if son 1 is colorblind, means both the alleles here is small c, X small c, X small c, and Y, right? Okay. Son 2 is also colorblind, means this X chromosome that son 2 is getting is also X small c. Okay. So, because it is present on the X chromosome, and sons will get their X chromosome only from mother, right? X chromosome in sons will come only from mother, means this X small c allele is coming from mother. Okay. But mother is going to have two X chromosomes, right? So what will be the other X chromosome in mother? It is clearly mentioned that two parents with normal color vision. Okay. So mother had normal color vision. So if mother has normal color vision, means the other allele on the other X chromosome in mother has to be X capital C. Then only mother can have a normal vision. Okay. Right. Now, in son 1, question is this two chromosomes, okay, that the son 1 has received, it has happened because of non-disjunction during which event? Okay. So, meiosis 1 and meiosis 2 you have to determine. So, in meiosis 1, homologous chromosomes separate. Okay. So, during meiosis 1, what will happen? That X capital C and X small c will separate. Okay. And in meiosis 2, the sister chromatids will separate. X small c here, X small c here. Okay. Now, because this disjunction was not happening properly, that is why both X small c has come in son 1. Okay. So, non-disjunction happened during which event? It happened in mother, and that happened during meiosis 2, where the sister chromatids were supposed to be separated, but they did not separate. So, both the X small c allele ended up in son 1. Answer will be option 2. Absolutely correct, Brinda. Vishal, did you understand the concept? Okay, so here again you see, for solving this question, you have to have a very good idea about cell division. Okay, in meiosis, what kind of chromosomes are separated during meiosis 1? What kind of chromosomes are separated during meiosis 2? Okay, see guys, it is very important for you to join the concepts of different units and try to see the whole picture. Then only you will be able to solve this kind of combination questions. Now coming to another combination type of question, which is here. Figures A and B respectively represent dideoxy sequencing gels obtained by partial sequences from 5' end of bacterial gene and its mutant with a point mutation. What type of mutation has occurred in the gel? Dideoxy sequencing gel. Okay, so dideoxy chain termination method of DNA sequencing is also called as Sanger's method, that you will have in unit 13. Okay, and you have to link it with the mutation here, right, in unit 8. So this question you will not be able to answer at all if you don't have any idea about Sanger's method. Okay, so like this kind of question, although this question came around 10 years back in CSIR exam, that time the number of this kind of combination questions were less. But nowadays, this combination type of questions have increased in proportion. Okay, because when your aim is to go for PhD after clearing CSIR exam, so you have to know the techniques, you have to know how it is applied for the basic concept. Okay, how to identify the basic concepts there. So it is very much important to develop your analytical skill. So for this question, you can answer it only when you have a good idea about Sanger's method. So Sanger's dideoxy sequencing gel, in that we get to know the sequence. So in there we will be adding DDNTP, dideoxy nucleotides. So when DDNTP is added, the chain elongation will be terminated. Okay, and then we will separate the DNA fragments, and each column will have a specific, it will have the sample from the tube that had a specific type of DDNTP, and then we will separate the DNA fragments in a gel. Okay, and then we read the gel from bottom to top. Okay, and what is this bottom? Bottom is like farthest from the well. So if this is the well, then bottom is farthest from the well. Now which fragment will be there at the farthest? The smallest one, right? That is the concept of electrophoresis. That smallest fragment will travel the maximum distance, largest fragment will travel the least distance. Okay, so we will read the gel from bottom to top, or farthest from the well to the closest to the well, and that will give us the DNA sequence from 5' end to 3' end. Okay, now how do we read the sequence? So wherever the band is coming, okay, so in whichever column the band is coming, that nucleotide is present there. So if it is DDTTP's column, so the last band is at T, that means at this particular end it is T. Next is in A, so next will be A. Next is G, it will be G. Okay, again G, sorry, not G, I'm so sorry, it is C here. Okay, and then C, then G, then T, like that. Did you all understand? Can I have a quick answer from you all? Because we are almost up with the time. So this is how we will write the sequence. Okay, so until and unless you write the sequence, you will not be able to identify which mutation has happened. So you have to write the sequence in wild type as well as in the mutant type. Okay, so then G, G, A, C, T, T, G, A. Okay, now similarly write the sequence for mutant type also. Okay, now when you compare this mutant sequence with the wild type, till here everything is same. Okay, and then from A to the end also it is same. Okay, but what has happened? That this G has gone missing. This G is not present here. Okay, so what kind of mutation has happened? G has got deleted. Okay, now do you have deletion here? No. Okay, but deletion, a single nucleotide deletion will result into what? It will result into frame shift mutation. So answer will be option C. Nonsense mutation, it is conversion of a coding sequence into, coding codon into a nonsense codon. Missense is where the codon is changed so that the amino acid is changed. Transversion, purine is substituted by a pyrimidine or vice versa. Now, all of this are base substitution mutation where a base is substituted with another. Okay, but frame shift mutation is the only one which can be resulted from deletion. Okay, so which mutation has happened? Frame shift mutation. Vishal, I'm sorry, did you not understand? So for this question, you have to have an idea about dideoxy gel sequencing. Okay, Sanger's method of sequencing, which is also called. So with using that concept, first you have to find out what is the sequence that you are getting in wild type and in mutant type. Okay, and then when you compare the sequence, so you will find what mutation has happened. Understood? Okay, great. Okay, so everybody, if you have any confusion here, so please go through the concept of Sanger sequencing, see how the red gel is read, how the sequence is analyzed. Okay, and then on the basis of that, you can decide what mutation has happened. All right. So with this, we come to the end of the class. Thank you everyone for joining the session. Okay, so as Shekhar sir says that dark cloud can never outperform a persistent sun, and that is you. So be persistent in your effort, keep practicing. This extra time whatever you have got is to utilize in the best way possible. Practice more and more questions. Keep attending the Brahmastra series every day in Biotechnica's YouTube channel from 6 to 7. And we will be coming with different topics every day. We'll be discussing the part C questions and we will be helping you. Okay. And also, we will be giving you the free paper where there will be extra part C questions along with their explanation. And this is for our Lakshya batch. If you are interested in joining the Lakshya batch, which will give you the boost that you need to clear the next CSIR exam, please go ahead, contact our toll free number, and we have some exciting offers also. And more about it, you can know on the toll free number. Thank you everyone for joining. It was a great session. See you all in another session. Thank you everyone. All the best. Keep practicing.